Fluorescence microscopy is widely used in biochemistry and life sciences because it allows scientists to directly observe cells and certain compounds in and around them. Fluorescent molecules absorb light within a specific wavelength range and then re-emit it at the longer wavelength range. However, the major limitation of conventional fluorescence microscopy techniques is that the results are very difficult to evaluate quantitatively. Fluorescence intensity is significantly affected by both experimental conditions and the concentration of the fluorescent substance. Now, a new study by scientists from Japan is set to revolutionize the field of fluorescence lifetime microscopy. Read on to understand how. A way around the conventional problem is to focus on fluorescence lifetime instead of intensity. When a fluorescent substance is irradiated with a short burst of light, the resulting fluorescence does not disappear immediately but actually decays over time in a way that is specific to that substance. The fluorescence lifetime microscopy technique leverages this phenomenon, which is independent of experimental conditions, to accurately quantify fluorescent molecules and changes in their environment. However, fluorescence decay is extremely fast, and ordinary cameras cannot capture it. While a single-point photodetector can be used instead, it has to be scanned throughout the sample's area to be able to reconstruct a complete 2D picture from each measured point. This process involves movement of mechanical pieces, which greatly limits the speed of image capture. Fortunately, in this recent study published in Science Advances, the aforementioned team of scientists developed a novel approach to acquire fluorescence lifetime images without necessitating mechanical scanning. Professor Takeshi Yasui, from Institute of Post-LED Photonics, PLED, Tokushima University, Japan, who led the study, explains, our method can be interpreted as simultaneously mapping 400 light stopwatches over a 2D space to measure fluorescence lifetimes, all in a single shot and without S. One of the main pillars of their method is the use of an optical frequency comb as the excitation light for the sample. An optical frequency comb is essentially a light signal composed of the sum of many discrete optical frequencies with a constant spacing in between them. The word comb in this context refers to how the signal looks when plotted against optical frequency, a dense cluster of equidistant spikes rising from the optical frequency axis and resembling a hair comb. Using special optical equipment, a pair of excitation frequency comb signals is decomposed into individual optical beat signals, dual-comb optical beats, with different intensity modulation frequencies, each carrying a single modulation frequency, and irradiated on the target sample. The key here is that each light beam hits the sample on a spatially distinct location, creating a one-to-one correspondence between each point on the 2D surface of the sample, pixel, and each modulation frequency of the dual-comb optical beats. Because of its fluorescence properties, the sample re-emits part of the captured radiation while still preserving the aforementioned frequency-position correspondence. The fluorescence emitted from the sample is then simply focused using a lens onto a high-speed single-point photodetector. Finally, the measured signal is mathematically transformed into the frequency domain, and the fluorescence lifetime at each pixel is easily calculated from the relative phase delay that exists between the excitation signal at that modulation frequency versus the one measured. Thanks to its superior speed and high spatial resolution, the microscopy method developed in this study will make it easier to exploit the advantages of fluorescence lifetime measurements. Because our technique does not require scanning, a simultaneous measurement over the entire sample is guaranteed in each shot, remarks Professor Yasui. This will be helpful in life sciences where dynamic observations of living cells are needed. In addition to providing deeper insight into biology. Perhaps most importantly, this study showcases how optical frequency combs, which were only being used as frequency rulers, can find a place in microscopy techniques to push the envelope in life sciences. It holds promise for the development of novel therapeutic options to treat intractable diseases and enhance life expectancy, thereby benefiting the whole of humanity. Story source. Materials provided by Institute of Post-LED Photonics, Tokushima University. Note: content may be edited for style and length. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.